Thank you very much for coming out on a Monday evening and listening to not one but two talks. And I hope to set up Matthias to talk about dogs. And I'm actually going to speak a little bit about dogs this evening as well, not too much. As I've just moved to Oxford 6 weeks ago, so we're very much in turmoil, and I left a very functional, working, very nice, efficient lab, and it's just pushed the blow up button, and now it's just chaos and confusion, and people are all over the place. And we'll get there. I think we're hoping by Christmas, but it'll probably be early in the new year, we'll start generating some results again. So yeah, I have to change my slide logo now, just the layout of my slides. So you're the first people to see this. And so of course, what better way to talk about domestication than to talk about pornography? Because the two have a lot more in common than you might guess. And the first reason for that is because they're both very difficult to define. And I think the way to really talk about this was from a famous US court case, Supreme Court case in 1964, where Justice Stewart, he was charged with saying whether or not a film that had been shown in an Ohio theater was obscene or not, and whether it was therefore classified as pornographic. So what he said was, trying to define what may be undefinable, we'll get to why in a second, obscenity laws are constitutionally limited to hardcore pornography. So he was trying to define if it was hardcore. I shall not today attempt to define pornography, and perhaps I could never succeed in doing so, but I know it when I see it. And this film is not. And that's the key point, is he's saying, you know, it's all kind of a bit mushy, it's a bit of a fuzzy thing. What are the criteria that you use to define hardcore pornography versus normal pornography, which is perfectly acceptable? I don't know how you do that, but I know it when I see it. And I think that's the way that most people think about things. And when we think of domestication, it's the same way. We don't know how we necessarily define domestication, but we know it when we see it. So when we see a wolf on one side, and we see a domestic dog on the other side, we couldn't tell you when during the process of domestication that some organism was now more domestic than wild, or now fully domestic, or whatever other terminology we'll use. But as soon as we get to dogs, we know it when we see it. And the same is true of wild boar and pigs, or just about any other domestic animal. We know what a wild boar looks like. We know what a domestic pig looks like. We know it when we see it. We don't. The process in the middle is a little bit more fuzzy. Same with chickens. Wild red junglefowl, which I'll talk about a little bit later some more. And in this case, you wouldn't even necessarily recognize them as the same two species. This is a domestic chicken from Southeast Asia, where it's mostly naked already, and they pluck a few feathers, and then it's used in fighting and a few other things. But the chicken can turn it from not just that. How about a Polish frizzle? You wouldn't guess that a Polish frizzle comes from a red junglefowl, but yet, same species, and how do you define when one becomes the other? It's difficult. And, of course, the silkie. I mean, that creature is just absurd. It should not exist, but it does. And to say that one is domestic and one is wild is no question mark, but where do we put that line down the middle is always the $64,000 question. And that's what we try and do. So we dichotomize. We say we have wild on one side, we have domestic on the other side, and there are innumerable papers that are written trying to put a nice dotted line between one and the other, and in between there isn't really anything except for kind of mushy goop. And you can put the line there, or you can put the line there, and you can write big papers that go on and on and on about why your predecessors put the line there, but you put the line there, and it's all just trying to put arbitrary decisions on a continuum, which makes it not really satisfying when we're thinking about the process itself. So another thing this leads us to do, I think, and quite wrongly, is that it allows us in a dichotomous framework to think about domestication as an event, because if you put a dotted line, you don't allow any process. You simply have a wild organism, and then you have a domestic organism, and then something happens in the middle, we're not quite sure what. And the problem I have with domestication event, and every time I see it in a paper, I just rage and I get pissed off and throw the paper against the wall. And I go to conferences and I'll say things like, we should never use this terminology. And then people in the room, who I know are in the room, 6 weeks later publish a paper using domestication event. And I send them emails, nasty emails. Why are you doing this? Because and the reason for this, the reason I get so head up about this whole thing is because it does a lot of things which I don't think that domestication really has anything to do with. Firstly, if it's an event, it implies some kind of human intentionality. And you hear this question all the time. Why did humans domesticate the cat? Why did we domesticate it? It's not, no, there's no why. Why implies intentionality, and that has nothing to do with early domestication, especially when we're talking about commensal animals, which I'll speak about in a second. Secondly, it means that domestication becomes technological rather than biological, and we remove the evolutionary aspects of domestication and start thinking of it as though we're inventing light bulbs, which, you know, has some play in it as well. But it's not just like there was no light bulb, then there was a light bulb. So it's not technological, and we need to get away from that kind of thinking. And thirdly, it implies that domestication can be invented by smart, goal-oriented people, which then necessitates the question, why? We did this because somebody around a campfire had a brilliant idea, saw a wolf, and said, you know what? I can make a small little creature with floppy ears and a piebald coat and upturned tail that will bring into camp and it won't eat our 2-year-olds. And we'll keep it around and we'll throw balls at it, it'll be great. Nobody does this. Why would you do that? Of course you wouldn't do that because all you have is a wolf to think about. So it must be something different than this process. There must be something else going on. And I have not a huge amount of time tonight, so rather than focus too much on that process, I wanted to give you a little hint about it and then take you somewhere else to tell you about some other aspects of domestication that I think are overlooked very often. And so just to give you a little clue about some of the ways that people have been thinking about the process recently, this is a figure that I drew with Joachim Berger, but based exclusively off of Mindy Zeder's work, which she published in 2012. This is all, I don't want to present it as my stuff. This is her stuff. And it's amazing, and it's absolutely fantastic. And what I think it really does is allow us to think about domestication in a very different and kind of interesting way. And what she says is that you have the wild animal on one side, and rather than just a dotted line, you then have a series of steps, some of which are intentional as you get closer to a pet, and many of which, at least early on, are not intentional and involve more of an evolutionary co-relationship between two different sets of populations. You will start with wild. You may end up with commercial breeds and pets and fully domesticated organisms. But in the interim, in the middle part, you have several different trajectories and pathways to get there. And the first one she describes is commensal, and there are a number of animals, including dogs and rats and mice and pigeons and lots of things where sort of get used to a human niche. And in that way, it's those animals that are kind of anthropophilic. They are taking advantage of the resources that we are providing and creating, and they're sneaking ever closer to that, to us and to the niche that we're creating, and we are maybe sneaking a little bit closer to them because we recognize that there is some value of having them around. They're not exclusively competitive or bad or negative in any way. And then from there you get to a habituation stage, which then leads to a kind of commensalism and a partnership, and very early on this is not goal-directed, this is not going anywhere, it's just happening, it's an evolutionary process, only after which people then are starting to put their own selective pressures for certain things involving different types of behavior or different sorts of phenotypes. But at least early on, it's very much to relate to organisms in this sort of evolutionary dance. The second kind of pathway that you can get from wild to commercial pets and breeds, but starts anyway, a very different thing. Cows, sheep, goat, large animals that are initially prey. And these are species that don't want to have anything to do with us because we eat them, so they're running away as fast as possible. We are going out, we are corralling them, and we are manipulating their population structures. And I would say in this case, it's still not an intentional domestication process. Domestication is an accidental byproduct of resource management. And the more that you hit a resource, the more that you manipulate it and shift it, the more it might not be there anymore. So then you you get caught in these labor traps. So you start taking out most of the males. So you bring up the number of females, but then they sort of mess with the whole dynamics of the herd. Then you say, well, okay, maybe this is easier if we bring them closer, and maybe it's easier if we start manipulating the way that they reproduce with each other. Now you find yourself in a situation where you're taking care of their entire life history on a 24-hour-a-day basis, and you never intended to do that in the first place. It's a series of small steps. So it starts off as prey, game management, herd management, extensive hunting, breeding, intensive breeding, and then you get to your pets and your commercial breeds. And the third one is this directed pathway, and this is a hamster, this golden Syrian golden hamster from 1930. It was plucked out of the desert, and in this case, this is absolutely deliberate. It is absolutely intentional. There was a guy, he was near Aleppo, he pulled out some hamsters from the ground, brought them to a lab 70 years ago, 80 years ago now, and now we have several million hamsters all over the world as a result of this. My grandparents were born in a world without hamsters. Just food for thought. So in that way, but that has colored our appreciation for what domestication really is. And in that way, so Dorian Fuller and I have a paper coming out, I think this month or sometime in the next couple of weeks anyway, where we look globally and we say, okay, show in every situation, zooarchaeologically, what are the first animals to appear in a domestic record, and what are the next ones, what are the next ones? And regardless of where in space or time that we look, the first animals are always the commensal animals. And that makes sense. You don't pick a gerbil or a hamster out of the desert and bring it into a lab unless you already have a model for what a domestic animal is. And in order to get to that domestic animal, you have to have this evolutionary process that happens initially. So dogs in one place, guinea pigs in South America, it's always the commensals that start. Then it's the prey species that follow from there, and then it's the directed species after that. The first directed species are horses or so, about 5,000, 5,500 years ago, where you're actively going out and manipulating organism and bringing it back into camp. The interesting thing, though, and the directed ones are in blue, something like 80 to 90% of all animals now on Earth that are domesticated are the result of a domesticated pathway, or sorry, of a directed pathway, which means that colors our view. That's what everything is, so that must be how it started, and that's not the case at all. And so it gives us a completely wrong impression of the whole entire process. So what I wanted to tell you about tonight, though, rather than the process itself, was kind of move a little bit and talk about other ways that I think that we often misrepresent and misperceive domestication. And this is also a little bit of a mea culpa, trying to make up for my role in making sure that everybody got the complete wrong end of the stick when it came to domestication. This is a paper that we published 2005, so next year will be the 10-year anniversary, and it's my most heavily cited paper. It's pushing 500 citations or something insane, and I'm kind of embarrassed by it because it's actually wrong. Wrong in a very dangerous way, actually. And so every time I get cited, I go, oh, that's great, but... so what we did was we got a global sample of wild boar and a number of domestic pigs, and we just looked at a mitochondrial fragment. So this is a non-recombining locus that's key for a reason to come up with. And we decided we'll build a tree from all of the wild stuff, and then we'll see how many domestic animals in different places in the world match those same signatures. So the first thing we found was that all of these different clades represent very well supported, highly differentiated, geographically and genetically distinct populations of wild boar, and those 7 circles are instances within those clades you will find domestic pigs that share either the identical or very closely related mitochondrial sequences, which we then knee-jerk interpreted to say, aha, those are happening in those 7 places in the world, so therefore pigs were domesticated 7 times, because it's very easy to draw the inference that if you have a wild boar with the same signature as a domestic pig, and that's different than somewhere else, then those domestic pigs must have been derived from that wild boar in a nice linear fashion, where that population was domesticated to domestic, wild boar were domesticated into the domestic pigs we know today. We did say in the paper, I've gone back to look at it recently, we were covering our asses a little bit by saying this is just a non-recombining locus, it's just mitochondria, and there are other ways that an organism could acquire a mitochondrial signature of a local wild boar population without necessarily being independently domesticated. But we just kind of, but never mind. That's possible, but it's not really something we can even look at with the data that we had, so we just ignored it, which is the classic scientific mistake. So I was full of bravado. I was only 2/3, 3/4 of the way through my PhD. It was a first author science paper. I was like, hey. So I just got high on the whole thing. And one of the things we said in the paper was the genetic evidence presented in this study provides clear proof for multiple centers of domestication across Europe. And oh my God, that is so disastrously wrong. I'm embarrassed to put that up for you. Firstly, never use the word proof. It's just insane. I just can't believe that the reviewers didn't pick up, that my co-authors didn't pick it up. I was a PhD student. I didn't know. I should have known. So that's ridiculous. And of course it doesn't, and it's wrong completely. I went even further in the press release, because we were like, oh, write a press release. Okay, that'll be fun. Our data show that domestication was not as rare as previously thought, and that the question now is not where were pigs domesticated, but rather where were they not domesticated? It pains me to put these up. The New Scientist picked up the story and they said 7 times lucky for pig domestication, pig tamers. This could not be more wrong, and I want to tell you why. So, well, in 2010, so 5 years later, I'm still hanging on to this paradigm. I'm still kind of desperate for it. And so we published a paper where we found that there were pigs in Southeast Asia that had the same mitochondrial signature as some domestics there as well, but there was no corroborating archaeological evidence. So still desperate to this idea that every time you found this correlation, that meant some sort of an intentional or independent domestication process. I decided the way to make your name in academia is to coin a phrase and to have that gain currency, and then people know you by your jargon and your lingo, and that gives you a long-term credibility. Well after you retire, people still using the language that you invented. That's much better than any paper you've ever written. So I coined cryptic domestication, cribbing off of some other things, cryptic speciation, etc., in which I said instances of which domestic animals share mitochondrial affinity with regionally differentiated wild boar, but where there is no corroborating archaeological evidence. And what you've probably guessed from this is what it's seriously intimating or seriously expecting is that it's not the fault of the DNA, it's the fault of the archaeologists for not yet finding the evidence to support what we consider to be independent domestication. You see, we're using language in a subtle but nasty way. This is, use your powers for good, not for And so a couple of years later, we published this paper in 2013, and it was actually through this paper that this light started ringing, or the light started flashing and the bell started ringing, and we started thinking, wait, maybe there's something else going on here. And this was really Joachim Berger who came up with this, much more than me. And this star that you see there, this is a chart showing ancient DNA through time and space across Turkey, through about 15 different sites or so. And you can see that there is a lot of change mitochondrially, both through time and through space. And that star surrounds the single sample that we got to work from the archaeological site, where all archaeologists are convinced is the very first, earliest evidence for pig domestication, independent pig domestication in Western Eurasia, almost certainly following some combination of a commensal route, possibly a little bit of a prey route as well. But it's certainly, it's like 2,000 or 3,000 years of slow evolutionary change morphologically as well. It's not just an intentional thing poking it out of the desert. And then what we find is that there are clear domestic pigs, morphologically, in Western Turkey, where they have a yellow signature rather than the blue signature. But there is no evidence there for the same kind of process that's happening at the sites in southeastern Turkey. So what Joachim said was, we're looking at this, and he's like, well, maybe what happens is, they were talking about small populations, people take their blue pigs, their blue domestic pigs that they domesticated in this place, move over to Western Turkey, and when there's a population of wild boar that is genetically and geographically distinct, they're yellow, not blue, and maybe those domestic pigs just mate with the wild boar that are there and acquiring that mitochondrial signature. Now, what that requires, because it's mitochondria, is that you have to get piglets born from wild sows, which is maybe a little bit difficult, certainly much harder than getting ones where the males can sire your domestic pigs, but not out of the question, because it turns out Mindy Zeder's also publishing some data from these sites which show that there are a ton of neonatal pigs. It's like people know when the wild boar are about to give birth, they follow them, and because they can't shiver, they have to build nests, and you know where those are, so you go out, you kill the mother, and you've got a whole litter of pigs there now at your disposal. You can bring them back and eat them, or just incorporate them into your domestic stock, and if one of those happens to mate with your males and give birth, now you're acquiring the mitochondrial signature. So what this is looking like is a mechanism for acquiring local mitochondrial signatures without being domesticated in the same way that we think about the process. And this, I should have known this, because in 2007, we demonstrated something very similar that was happening in Europe, where all the red pigs in Europe, or wild boar, were being swamped by domestic pigs coming in from Western Europe, from Western Turkey. And in this case, all the little yellow dots, we did this double blind, were domestic pigs, and all the red ones were the wild boar. So people are moving pigs domesticated in Turkey all the way to Paris, and then by 3,500 BC, you get a lot of yellow and a lot of red there, but then very soon afterward, from 3,900 to 700 BC, in Europe goes completely red. There is no residual Near Eastern mitochondrial signature that lasts beyond 3,500 BC in Europe. So what's happening there? It's not that the local wild boar in Europe are being independently domesticated, it's just that the mitochondrial is leaking from the wild population into your domestic population through whatever mechanism. But that's very different than an independent domestication process. So in that way, there is no such thing as European domesticated wild boar. It is just acquiring, through gene flow, all of the mitochondrial signatures from that population. So we should have known, or I should have known. It just didn't hit me. And we know that there is a whole lot of evidence for admixture, but we tend to think of it as a very recent process. So this is a brilliant paper written by Mirta Bosse out of Wageningen earlier this year, where by looking at full genomes of East Asian domestic pigs and European domestic pigs and wild boar, they're able to find that when people started doing these things called improved breeds in the 19th century, they were deliberately bringing domesticated pigs from Asia into Europe. They were then selecting for the very traits that made those Asian pigs better. So they had, I think, more offspring, and they had more nipples, and they were longer, they had more back fat, and all kinds of things, and that allows you to find the genetic mechanisms that underpin those processes. So we know this integration is happening, certainly in the 19th century, but probably a lot earlier either. And then I started doing just a kind of a general search through the literature, and this was another paper that I worked on in 2008, where we demonstrated that, yes, most chickens are, or all chickens are, mostly derived from red jungle fowl. But there is this yellow leg trait that actually comes from gray jungle fowl. So there is some admixture here. It's not just that domestic chickens are from one wild ancestor. They're from at least two wild ancestors, and two other wild ancestors may have contributed some genes as well. So kind of a strong hint already, a flavor of admixture happening. And then you start plowing through the literature and you realize, holy shit, it's everywhere and it's everything and it's all the time. It's insane. So, cows. You have taurine cows from Western Eurasia, you have Indocene cows from South Asia, you have hybrids here on the bottom left, and every single cow in Africa, every population is some combination of those two. It's never 100% or 0% of one or the other, it's always some split. Different proportions in different places, but it's always a hybrid of some sort, all the way throughout the entire continent. Horses. The spread of horse domestication out of Western Eurasian steppe was characterized by high levels of introgression from local wild populations. People are using different terminology but saying the same things. And as soon as they have the genomic techniques to start looking for admixture beyond the non-recombining mitochondrial locus, they start seeing it. It's like you just open that window and the whole thing comes washing in. And that was, I know another paper that's coming out in a couple of weeks in PNS that I've just seen that's massive amounts of admixture in early horses as they were being domesticated with wild populations all over the place. Bees, not a surprise perhaps, but still interesting. We found that managed honeybees actually have higher levels of genetic diversity compared with their progenitors in East and West Europe, providing an unusual example whereby human management increases genetic diversity by promoting admixture. So in this case, you have a population that was domesticated from a wild species. That domestic population then starts hybridizing with a whole bunch of other wild species that were not involved in the original domestication. And so your domestic population now incorporates everything, and it's just a big melee mess of a whole bunch of different stuff with a lot of genetic diversity in it. Apples. Bidirectional gene flow means admixture. It's all the same thing. Between the two domesticated apple and European crabapple, resulted in the current M. domestica being genetically more closely related to this species than its Central Asian progenitor, M. sylvestri. Now, if you think about that, that's exactly the same story as the pigs. Domesticated outside of Europe, brought into Europe, and very slowly starts to mimic everything that's in Europe. So that the apples and the pigs really are sharing very similar evolutionary trajectories. Grapes, sure, same exact story. Find support for a Near Eastern origin of vinifera and present evidence of introgression from local sylvestris as the grape moved into Europe. If you get to a place and you can hybridize with something, regardless of whether that thing has been independently domesticated or not, you will. And that's just what happens. It's just biology. Everybody's shagging everybody all the time. Rice. Protracted domestication process finished around 65 to 6,000 years ago in China, and about 2 millennia later in India when hybridization with Chinese wild rice took place. And there's all kinds of papers about African wild rice now contributing genes to domesticated versions. Everywhere you go, if there's some kind of rice variant, it will have contributed DNA to the rice that we know in that place. And this is a paper just out a couple of weeks ago, and I just love this. Because I saw this and I just kind of blew my mind. I was like, this is perfect. Like, I can just stop doing this talk now. Because this is citrus. So this is oranges, tangerines, tangelos. I never know what the difference is, but all that stuff, right? And this is just great. Okay, we'll try to go slow here, but it's everything. Although cultivated pomelos represent selections from one progenitor species, Citrus maxima cultivated mandarins are introgressions of maxima into the ancestral mandarin species Reticulata. The most widely cultivated citrus, sweet orange, is the offspring of previously admixed individuals, but sour orange is an F1 hybrid of pure that and pure that, thus implying that wild mandarins were part of the early breeding germplasm. Moreover, a Chinese wild mandarin, they don't even know what to call it, diverges substantially from C. reticulata, thus suggesting the possibility of other unrecognized wild citrus species that are, guess what, throwing their stuff in with the lot, making just a giant melee of just orangey colored fruit, which is just all over. And if that rings a bell, well, it should, because this is us too. This is the most recent version that is out of David Reich's lab at Harvard, Kay Proofer's paper from earlier this year, where this is modern humans, Denisovans, Neanderthals, or at least Denisovans, we didn't even know existed until a couple of years ago. So humans, Denisovans, Neanderthals, all swapping genes, and these are not insignificant levels, several percent each, and even more so, a potential unknown hominin, which is it's like a genome in search of a fossil is the way I've heard it described, also contributing DNA to one of these lineages. Everything is mixing all over the place all the time. When we look at the single non-recombining locus, it looks nice and bifurcating, but when we look at other stuff, everything just comes back together again, and just a melee of a mess of a non-tree bush thing. And I think we're finally figuring this out. So this paper is a week old or 2 weeks old or something out of SK's lab. And what I love about this is that rather than thinking of these things as discrete entities, a bit like the oranges, they say instead of inferring a few discrete migration events from Asia into Europe, we now see evidence that humans in Western Eurasia formed a large metapopulation with gene flow in multiple directions occurring repeatedly and perhaps continuously. We like to think of our everything as you have a discrete this, discrete this, discrete this. They all have their independent histories, and then they all kind of admix in a nice predictable pattern if admixture takes place at all. And what SK is saying here is like, well, actually, certainly when it comes to humans, it's just and as soon as we start to say there's just one thing, you break that down a little bit, and you go back into the past, you realize that's made up of everything else, and that's made up of everything else, and everything is just a constant blend and mix of everything together. Which is problematic if you're constantly thinking about bifurcating phylogenetic trees, which is the way that a lot of people reproduce, certainly what I was doing when I was looking at mitochondria. So just to show you how true this is, I thought I'd give you a flavor of some new data that we've just generated on dogs. And this is a PhD student, my old PhD student, not my new postdoc, pulling all this together. And I just love this. These are both stuff that we've generated and stuff we pulled off the web. These are ancient dog samples going from anywhere from 100 to, say, 8,000, 9,000 years ago, from Oceania, from Central, from Europe, and then from North and South America. And what we did was, this was actually the idea of my, she's now just finished her first year PhD student, just unbelievably brilliant. She just had this cool idea, where she said, why don't we look at the correlation between genetic difference, distance, and geographic distance. The idea being that if you have a population here and a population here, and they are from different wild ancestors, then if you look at that correlation, there should be a very strong, even as high as 1-to-1 correlation between genetic differences, which would be very different from the two populations, and geographic distance because they're far apart. As soon as those populations start coming together and they start admixing a whole lot, especially if you're looking at mitochondria, then the genetic distance will still be great, but your geographic distance will be very small, in which case you lose that correlation. So you can have a perfect correlation of 1, which means that you can predict it very nicely. A correlation of 0 means that there's no correlation whatsoever between the genetic and geographic distance, and that red bar represents modern dog populations globally, which is interesting, because what that suggests, and it's no surprise, that people have been moving dogs all over the place all the time, and we can take it one step further with a separate analysis using AMOVA, where we're able to demonstrate if you take 20 dogs from any breed, non-breed, geographic location anywhere, they will have not just the same haplotypes, but the same proportion of haplotypes as 20 dogs from anywhere else. Everything is just a giant blended soup, but that's modern. What happens if we go back and look in the past? And it looks like this. And it's so amazing. It's super cool. So going back 9,000 years ago, it's not quite 1, and the error bars are pretty big because the sample sizes are small, but we have a definite trend here where back in time, we had very strong correlation. When you have dogs from different parts of the world, they are exemplifying very different and highly distinct genetic differences and geographic distances. But through time, people start moving them around, and you start losing that correlation because you're starting to blend and mix and move and distribute, and it starts to become much more of a homogeneous soup. And the crazy thing for me, when Lisa first produced this, was you look here, when it drops precipitously, really quickly all the way down, that's exactly the same time as when people are moving pigs during the Neolithic into Europe, thus doing the same thing, reducing the difference, reducing the correlation between your genetic and your geographic distance, because now you're mixing a bunch of mitochondrial haplotypes from all over the place. Wow, that just blew me away. I was like, holy cow, this is happening 9,000 years ago, 7,000 years ago, much less yesterday, and 50 years ago or 100 years ago with the kinds of improved breeds that we're talking about. So what? So big deal. Who cares, right? Except that I think what this really demonstrates is that a bit like the pornography definition, we need to be more exacting about what we're saying. And rather than use the word nuance, which sometimes people jump out of, do you have this experience where you use the word nuance? Like, oh, you're academic, you're always talking about nuance. Okay, fine, no nuance then. How about sophisticated? Makes you sound smarter, makes you sound better. I think it's a greater appreciation for the word domestication, which means we need to differentiate between the independent initial process, regardless of which of those 3 pathways that I was discussing before, and secondly, subsequent admixture with populations that were not involved in the original domestication. That will give us a much better, deeper impression for the entire history of domestic animals, which means we can say populations of wild boar in eastern Turkey were domesticated, but populations in Europe, wild boar, were not domesticated. There was something else going on through the admixture. And what Dorian Fuller and I have come up with is trying to put a new term out there. We've called it introgressive capture. Yeah, I know, it's not great. It's the best thing that we can come up with. If you have a suggestion for another way to sort of, what's a word that we can use other than domestication to mean grabbing genomes from things that weren't involved in that process, please let me know, because it's far too long and it's a bit of a mouthful. And lastly, I think what this means is that when we don't use this nuance, when we don't think about this sophisticated difference between these two things, it leads to a significant underappreciation of admixture, which then confuses definition and biases interpretation making it very, or certainly much more difficult to infer the past from the present, and I think means that we are myopic when we try and then think about the processes that led to domestication. We start asking questions like, why was the cat domesticated? Which is just a completely wrong question to ask. So what? The way to think about this more generally is that if this is your modern population, and this could be anything domestic, it's just a giant bowl of tomato soup. And there's a lot of ingredients that go into tomato soup. And if you have the ingredients, you can make the soup. But if you start with the finished product, how do you predict which ingredients and which proportion of those ingredients and what order those ingredients went into to create that soup? That's a much more difficult process, and that requires large amounts of genomic data rather than single locus, although single locus is great to compare the two as well. And it requires, I would think, some ancient data so you can actually see the timing from when these different ingredients were coming in and playing a role in the creation of the modern genomes. Because working backwards from soup is a very difficult thing to do. So just on the plane, as I was getting off, it occurred to me that rather than just finish on soup, I thought I'd finish because I've come here to Switzerland today. And it's this kind of, first of all, raise show of hands, how many people are actually Swiss? Okay, so fair proportion. I was thinking maybe it was a big, important university that a tiny proportion of people would actually be from here. I know that's certainly true of Oxford, where every accent I hear is American. So Switzerland, I think, encapsulates this perfectly, because the Swiss is one nation, it's one flag, and you're probably about to win the biggest tennis prize in the next week, assuming Fed's back gets better. But in the meantime, this is just a map of all the different languages in Switzerland, and I'm sure if we were to map on any sort of a cultural affiliation or genetic heritage or anything else, it would look equally sort of disparate and all over the place. And I feel like because it's sort of right in the center of Europe, and there's Neolithic migration pathways coming in through the center of Europe and also along the Mediterranean, and the Swiss region is this giant melting pot of everything coming together. This is the country encapsulation of all that admixture that I was speaking about for the last half hour. It's sort of the perfect place to be discussing this, really. So on that note, I'll finish up, and thank you for your time. Question now, questions later?
Now would be good.
Now's good.
Thanks very much indeed.
Sure, Dasha.
I was wondering, if, in addition to the mitochondrial DNA, are you now looking into the Y chromosome as well? Are you incorporating other autosomal markers?
Well, we are now, with all the projects that we have currently in the lab, are taking advantage of all those wonderful capture techniques to be able to mine out the genomic data that we are most interested in, the genomic sequences that we want. And we will probably include, may include a few Y chromosomal markers on there. I'm kind of, unless there's a specific question involving somehow male dispersal or something, I'm happy to sort of dispense with that and go with a combination of the mitochondria and the nuclear in order to combine the two. And the thing that I want to like to stress, actually, is that, and I've read some papers about this, and I've seen some reviewers say, oh, this is mitochondria, it doesn't tell you anything. We're moving beyond the mitochondrial genome. Bullshit. We're not moving beyond the mitochondrial genome. That's like saying, because you now have TV, I'm going to get rid of my radio. You don't know. There are two different things that have, two different markers that have very different evolutionary histories, and they're both interesting and useful. And when they tell the same story, then that's great. When they tell a different story, that's even more interesting. But you need two in order to tell you more than one story. So we are now going to layer the nuclear story on top of the mitochondrial story and look at dogs and pigs and chickens and a couple other animals to boot. Whether or not we include a large number of Y chromosomal SNPs kind of depend on how many separate markers we can go for with this capture approach. But not out of the question, but I'm kind of more tempted just to try and get a larger picture of a full genome perspective. Yeah?
When you looked at the correlation between geographical distance and genetic distance, it went down in general, but there was also incidents where it went up again. How do you explain that? Would that be local adaptations again that are showing up? Or
yeah, I think that's certainly true. It's also, I would like this data to be true. We haven't published it yet. And one of my colleagues was looking at it and saying, your error bars are big enough that I wonder how much of a significant difference there is between the top part and the bottom part. And it may very well be that those little fluctuations that you see in the middle there are not reflecting anything important, but just could be noise of the sample size. And it's interesting there because there's a whole lot of samples in the last 500 years and a ton of samples in the Neolithic. And where you see it start to go up and the error bars get really big, for whatever reason, nobody's interested in Roman dogs. Like, nobody gives a shit. This is weird. Like, there's a whole span of time where their sample size just drops considerably, which is the beginning and the end, but in the middle, man. So I think it's probably more of a reflection of the noise of the sample size than anything else. But if that is real, then I think maybe something like local adaptation or just accumulating mutations in that space, which may be differentiated again in a lack of gene flow, could be generating that. In the same way that there's that great John November paper from 2008 in Nature where they recapitulate the map of Europe on a principal component analysis by looking at SNP data from human populations, so that it looks, if you just look at the PCA, it looks just like a map of Europe. And the reason that that looks so good is because every single sample on that map, the person had to confirm that all 4 grandparents were all born within 20 kilometers of each other. Now, I don't know if that's true of anybody in this room. It's certainly not true. Is it really? That's impressive. I'm sorry to hear that. It's certainly not true of me. And so if you think that if you were to incorporate data from people whose grandparents lived a little further than 20 kilometers from each other, it would just be the kind of soup that I was talking about at the end. So, and I think that's probably much more likely with dogs than it is with people. And that really speaks to the fact that the medieval era was a time, an unusual time over the last several thousand years of human history when people were a lot more settled and there was a lot less regional admixture than there had been for millennia before that. But that, again, colors our impression that people go to one place and they stick there and then they become that thing and then they hybridize with that thing. Never. That's a good question. Yeah.
I really like the genetic soup. But in the end I was puzzled how many domestication, sorry for using this obscene word, events were with the pigs.
So what Joachim and I are suggesting is that what this implies is that for every single animal you should start with the supposition that there was one and one only and everything else is subsequent admixture. And the onus is on you to demonstrate something truly independent because admixture is so pervasive. With pigs, there's no question that it's 2. Because you have wild boar in East Asia that are contemporaneously with the ones in Southwest Asia being domesticated from an independent wild boar population, and that's happening in Jiaohu in China at the same time that it's happening in China, Tepesi in Turkey. So it's hard to suggest that those 2 would have any kind of communication and there was something going on there where there was not independent. Beyond that, though, I think the incorporation of wild boar from the whole rest of Eurasia is subsequent admixture. So I'll give you 2 for pigs, but I'm not even entirely convinced that there's 2 for cows. I think that there's certainly the possibility that Indocene cows from South Asia look Indocene with a hump only after taurine cattle that were domesticated in the Near East were taken over.
Can you somehow collaborate with the archaeologists on that point?
I have been doing, yeah.
I remember that it's about 30 years ago when I was in prehistoric archaeology. The discussion was that there were 2 domestication events for pigs, so what you are saying. So I mean, it would make a kind of independent proof.
Yeah, no, and we've discussed that. Yeah, we put that in papers and things. With regard to the cows, I don't know if you know Richard Meadow at Harvard. We've been discussing it with him, and he's open to that possibility. But what everybody right now says for cows, there's 2, and then there's this possible 3rd one in North Africa. No, I don't buy it for a second. Only because now you can see that I've gone. You know, I resisted change. It's like, and then as soon as the light popped and I was demonstrated how wrong I was, I went, okay, I'm over here now. And all those guys are totally wrong. I don't like them at all. And anything in the middle, it's very much Thomas Kuhn's structure of scientific revolutions. You know, where you're standing in one place and you're waiting, and then there's a paradigm, and there's like, I kind of like this paradigm, and all this negative data is coming in and accumulating. You're like, no, I can bat it all away, and then you go, okay, screw it, and you go over here. Okay, fine, now I'll accept all that. And now it makes a lot more sense in the new paradigm, and that's how change happens in these kind of big jumps rather than a very gradual process. And we all like to think. I would certainly like to think that, well, I take out a bit of new data and I will shift it. No, clearly. I mean, I'm writing bad press releases, hyping my own stuff, and it's all completely wrong 10 years out. So God knows what I'll be saying 10 years from now if you have me back.
On that line, you now start a new job in Oxford, and with a big lab and so on, and you have your ERC grant and so on. And it seems to me there's a lot of potential and wonderful things that you can find out with this molecular studies in conjunction with archaeological materials. So what would be the dream? What's the big thing that you expect to taste, or where do you see yourself in
Is this an interview?
10 years or 20 years? What's the potential, or what's the thing that these domestication studies will tell us about human evolution or human, and I don't know, something, a vision for the future, or what can be accomplished? Or
how long you got? Well, I'll try to do that in 30 seconds.
No,
So I think that if you'd asked me that question 5 years ago, I certainly was not at the forefront of the sequencing revolution, and when I did my PhD, by the time I finished my PhD, sequencing prices had dropped by not even yet an order of magnitude. I mean, it was a relatively small thing. And I didn't foresee how the sequencing revolution could be applied to paleogenomics, and we didn't have the word then, and looking at archaeological material. So asking me to look 5 years from now, or 10 years from now, is just like, I have no idea. What it looks like what's happening right now is that there is a massive rush to sequence the bejesus out of absolutely everything ever, forever. And there's just a massive data collection exercise. In fact, Svante Pääbo's most recent paper in Nature, which was end of October, 45,000-year-old human from middle kind of Siberia. And if you look at that paper, Svante is amazing. He's coming to Oxford next week, and he does brilliant work. And I could sort of sense his frustration with this, because they had a high coverage, but 20-30x coverage genome of a 45 oldest human that's ever been sequenced. I mean, all this great stuff. It's in Nature, and they said nothing. There was no story. It must have been really terribly frustrating to have all this great data. So they just hung the paper on a mutation rate thing, which is like, all right, whatever. But the title is just like the sequence of a 45,000-year-old human. That's it. It's not like it shows us this, demonstrates this, no, nothing like that. It's just, well, here you go, here's some data. That's all of it. And I think that the field right now is still a little bit immature in that sense, in that we are in a massive rush. We're not really thinking about the questions. It's just like, let's just sequence everything and let's throw a ton of money and just see what we get. We'll throw out all the bioinformaticians and deal with the questions later. And that will continue for a couple of years to come. But we'll get to a point where I saw one of the science papers, News and Views, said something like, another week, another ancient human genome. And I was like, yeah, big deal. Who cares? Which is amazing when you can think where we were 5 years ago. So what I think is the key thing, though, it's the questions. It's what I'd love to do with dog domestication, and what we're trying to do now as part of the ERC is to really definitively answer this question: how many wolf populations went through this commensal domestication pathway, and how many of those wolf populations subsequently contributed DNA to dog populations that were coming into that area? And I don't know what that number is, because wolves are everywhere. And I think a lot of dog domestication papers tend to look at the dogs and not the wolves. And if you're not looking at the wild ancestor, and you're not looking through time, when we know there's all this admixture, it just seems like we're missing a very large chunk of the story. So there's a lot of really cool stuff that's going to be done, even if we have to wade through genome after genome after genome of stuff to finally get there.
Somebody will pose a question, but what you're saying about the collection of data with the genomes reminds me of what went on in morphology probably 200 years ago, because at the end the anatomy was not recorded, so there was this phase of just documentation. And I have a friend, Kurako from Japan, he talks about doing a natural history of the genome. And a lot of it must be exploration in this regard. Maybe
Sure.
there's some parallels
Absolutely.
in the development of this field as it has been in other fields. But somebody had a...
I just had a comment on exactly that. I'm a human osteologist, and it's exactly that. Getting data for genome sequencing and isotopes. Isotopes
Sure,
that's the big hit, and you just need some isotopes in whatever you're doing, and otherwise it's just not worth anything. And sometimes I think, yeah, but why? I mean, is it really the question I want to ask? Or is it just because everyone does it right now?
I would go one step further. I'd say, is there a question?
Yeah, exactly.
I had people come up to me and go, I have the sample, you can do the DNA. I'm like,
Yeah, for what?
why?
Yeah.
Give me a rationale. Pitch me an idea here. Don't just say, it's not a service. You don't just generate a sequence.
No.
Say, here you go. It's got to have some purpose.
Yeah.
Right? At least you would hope so. Even if it can be exploratory. I mean, if you don't know anything, you got to start blind. You got to walk into a cave with your eyes closed and feel out where the walls are and see where the blind alleys are. I mean, that's absolutely a part of science. But if you have the methodology where it is relatively well worked out, and you have a sample with context, and you've got a hypothesis about something, why not incorporate that into the sorts of stuff that you're doing? And I think most people do. But sometimes it's easy to say, oh, let's just do the isotopes. Let's just get those values. What are you going to do with them? I don't know. We'll plot it. What are you going to do with the plot? I don't know. We'll publish it. Great.
It's very sad.
I don't know.
Any other question? All the molecular stuff that you've been talking about refers mostly to phylogeny, reconstructing the relationships of things. Do you see much going on or much potential in the studies that look at the genome, but as more as it relates to the generation of phenotypic disparity in these different breeds and so on?
Sure.
So how do we explain, or how what is there something we can learn from domestication to help us understand the origin of morphological diversity?
Absolutely.
So do you do anything in your lab, or do you know other labs doing it?
So I did that for a couple of years in Sweden with Leif Andersson, which was looking at functional genomics and trying to work out the genetic mechanisms that underlie specific traits with a larger view toward understanding general things about domestication. Are domestic animals just kind of hyper-selected for standing variation, or are there new variants, new genetic variants that are popping up as part of the domestication process that we then cherry-pick and really accentuate in the population? And I think that debate has gone back and forth a couple of times, and I'm definitely now leaning in animals toward standing variation on a lot of separate traits that each of which individually only represent a small proportion of the overall morphological variability. For things like behavior, for things like growth. Having said that, there are times, relatively late in the domestication process, when a new variant will pop up that is not part of the standing variation, where people go, ooh, that's cool. And then it becomes like a Jones's thing. It's like, well, my pig is not just black, but I've got this white banding on it. And everybody goes, oh my God, he's got a white band. Okay. And so then you go and you grab the pig with the white band and you selectively make sure that that's the one producing more offspring than all the rest of them. And then now everybody's got the new one with the white band. It's like the Sneetches with the stars on their bellies from Dr. Seuss, anybody? There's some Sneetches and they have stars on their bellies, and stars without stars on their bellies, and without the stars. And it's about fashion and about what you consider to be important, and that is shifting all the time. And I think our domestic animals represent that. I mean, all you have to do is look at shifting patterns of popularity of dog breeds to know that how strong selection can be over a short period of time, and sometimes to the rather disastrous consequences of the breed in question. It's almost like we tend to focus in on something and then accentuate it into oblivion, and then move away and then start doing it on something else. And we've been doing this not just with domestic stuff, we've been doing this wild stuff for a long time too. For a little while I was collecting, probably should still be, every time there's a white or albino version of a wild animal, it pops up on the BBC as like a special thing. So there was a white stag in Scotland, but they wouldn't tell you where it was because they knew somebody was going to go out and shoot it. And I just love that. And there was a white whale in New Zealand, and it was the same thing. Well, we can't tell people because somebody will go out and shoot it. And it was like, that's just kind of interesting. That we like it so much that we have to go out and kill it.
Well, maybe it's time for a break, get a drink. You are welcome to bring your drink back to the room so we can start at 10 minutes of the second. So thank you very much.
Right, thank you.
Thanks for